All right. Welcome, everyone, to the webinar 3, the last webinar of today's Pause for a Cure. It's my pleasure to introduce our first speaker, Dr. Adam Resnick. He is the director of the Center for Data-Driven Discovery in Biomedicine at the Children's Hospital, Philadelphia. And I've had the opportunity and the pleasure to know Adam for a number of years, and he is a wonderful advocate for children's research, and it's really exciting to see him here at the Pause for a Cure event and talking about canine cancer as well as childhood cancer. So with that, I'm going to turn it over to Dr. Resnick.
Welcome, everyone. My name is Adam Resnick. I'm from the Children's Hospital of Philadelphia, and today I have the privilege of really presenting to you on behalf of consortia-based efforts, collaborations, and a NIH-based initiative called the Gabriella Kids First Data Resource Center. And my hope is to really go through both the evolution of some of our efforts, beginning with pediatric brain tumors, and then the deployment and implementation of platforms, how we look and think about interoperability of those platforms, and more specifically, how we hope those efforts can really grow in the context of canine-based comparative research as intersected with opportunities for discovery in pediatric cancer research. I couldn't really proceed without really being transparent about my own history and commitment to canines or dogs as part of my family ever since I was a child. As you can see, I've always had a penchant for large breed dogs, and also the probably shared belief of every single owner that despite statistical likelihood, your pet will be with you for a very long time. But unfortunately, like many dog owners, I experience the suffering attributable to cancer. Initially with my first dog, this is me as a graduate student, with Thomas, a St. Bernard, who only lived a few years and really succumbed to cancer. Geronimo was a Bernese mountain dog who was with us between 2007 and 2014. And now we actually have two really great, amazing pups, Padfoot and Navi, who are great Pyrenees. This is them in 2015, and this is Padfoot with my own 14-year-old here actually in 2019. So we are and always been great dog lovers. But really our efforts began in pediatrics with pediatric brain tumor, initially as a single laboratory and now as a full-fledged center here at CHOP, really focused on how to address what is the leading cause of disease-related death in children, which is pediatric cancers, and more specifically, some of the worst are pediatric brain tumors, and recognizing very early on that despite this appellation, it really is a rare disease context that requires rethinking about how to do research if you want to change any of these statistics associated with really the suffering and context of the disease that has unfortunately not wavered dramatically in the context of new drug development and therapies. A lot of this has to do with the need of really thinking about collaborative or multi-institutional research, and how do you support the collection curation of biosamples across multiple institutions? How do you support large-scale data generation, integration, accessibility, and use of those data? And here you can see really a timeline of those efforts that began in 2013 with 4 institutions in the context of the Children's Brain Tumor Tissue Consortium that has been recently rebranded as the Children's Brain Tumor Network, hence the CBTN logo here. But now today is more than 18 institutions strong. And along the way, we've had to implement a variety of different tools and resources that we think are equally applicable in the context of comparative biology research. And this is a uniquely opportune time to pursue this. We don't just collect specimens and samples and data for their own sakes, but really for clinical translation. And so early on, the Children's Brain Tumor Tissue Consortium, or CBTN, really began partnering with other consortia-based initiatives like the Pacific Pediatric Neuro-Oncology Consortium that together really represent more than 30 institutions across the United States, Europe, Australia, and Asia, dedicated to really continuum across biosample collection, large-scale data generation, analysis, interpretation, and clinical translation in the context of biologically relevant clinical trials. The biorepository-based efforts really now are fairly robust, with more than 3,500 subjects included or enrolled in the repository, more than 45,000 biosamples, and an initial set of about 1,000 whole genomes and RNA-seq across all histologies. We recently had the privilege of working with the NCI CBTech group on a pan-histology proteomic study, as well as a single-cell study. So really creating multimodal data for analysis of disease in ways that we hope then can drive new biological understanding and its implementation in the context of clinical trial development. To make this work across institutions asynchronously in a way that's engaging both scientific interests and clinical trial development interests in real time, you need platforms. And very early on, we both borrowed and stole and mimicked things that the NIH was doing in the context of development of cloud-based resources by partnering with Severn Bridges in the development of Cavatica, a workspace environment for analyzing genomic data. We partnered with the CBio portal team to develop Pete CBio portal, and more recently launched the Kids First data resource portal. Together, these are environments that provide for really across-the-board engagement of both data scientists, bioinformaticians, engineers, biologists like myself, in ways that you bring these multiple communities together on behalf of convergence research. To do this, you really have to have a modular approach that brings together these tools and resources in an efficient way across the workspace environment that support non-local access, use, integration of new data sets, portal environment that can empower users to discover those data sets across data modalities, knowledge bases that integrate process data for hypothesis generation, integration across molecular data sets and clinical and phenotypic data, a more recent effort that Alison Heath and our team has been leading with the NIH FIRE working group. And then really in the context of pediatric, where multiple institutes and centers generate data, interoperability across those data resources at the NIH, as supported by, for us, Strides in AWS, a program that really empowers cloud-based compute, storage, and analysis. This provides an end-to-end approach that not only transforms samples to data, to information, to knowledge, and hopefully impact through really an iterative cycle, but that double funnel of both integrating multiple different data types across multiple different samples and hopefully multiple different contexts of both human and non-human disease, and then also integrates at the other side the human element of discovery across different domains. This really came to fruition building upon the Kids First efforts building upon the brain tumor efforts with the launch of Kids First, which really was a unique program by the NIH that sought to intersect for the first time pediatric cancers and structural growth defects, recognizing that really development is a unique context for disease that might have shared biology in ways that could be approached through large-scale data generation. There's been more than 30 different studies to date with more than 30,000 whole genomes sequenced across a large number of very variegated diseases. And our hope is that this provides a platform to then further intersect in the context of comparative biology through approaching canine cancers. This, of course, should not be unusual to any of the audience, but I think it's worthwhile sort of noting this context of both structural or congenital birth defects and cancer is not unique to childhood. It's actually also present in every one of these breeds. Each one of these numbers is really representative of a congenital or heritable disorder, really representing the same type of intersection that we see in pediatrics. And several recent studies especially have noted, registry-based studies have noted this intersection with the risk of hazard or risk increase for children who have either cancer or structural birth defect for the other. It's within that landscape of both platforms, data sets, pediatric cancers, and especially pediatric brain tumors that we sought to really recently develop these further, really using the same approach we did in pediatrics, but leveraging them on behalf of new efforts in canines. So we partnered with Rebecca, Molly, Nicola, and the TGen team to really begin some new data generation efforts in in canine brain tumors across histologies. We're just in the beginning of these efforts, and at the same time focus on ways that these efforts could scale in the future by creating reproducible workflows in the Cavatica platform, leveraging what we've done for Kids First, but bringing it to bear in the context of canine analysis, such that those workflows with the appropriate genome builds can be reproducibly used for analysis and integration across data sets, not just those that we generate, but those of others. And that's been for us a recurrent theme, the need to really think about data that's generated across an ecosystem and bringing that data together. It's not uncommon for different data sets in the pediatric context to be generated under different programs in different institutes or centers, and the need to interoperate across those. We've had the privilege as part of an NIH Interoperability initiative to really work with NHLBI, NHGRI, NCBI, and NCI to really drive those interoperability across platforms, and our hope is to now extend this in the context of the integrated data commons. Indeed, you can do this now. You can go to the integrated canine data commons, select data sets, and bring them into Cavatica. Likewise, you can do this with, let's say, roles data in dbGaP. And very shortly, you'll be able to do this in a much more seamless way by simply taking your manifest of interest and imported the data into Cavatica in the very same way that you can do this now with the Cancer Genomic Cloud, in part leveraging our partnership with Severn Bridges. This provides for users the capacity to then take these diverse data inputs, whether it's data that we newly generated with partners, existing data sets that have already been published on, or that are in the part of the Canine Data Commons, and begin to intersect them with the pediatric context. Looking for that intersection across either pediatric brain tumors and canine brain tumors, or even expanding beyond that particular histology as we move forward. And then taking those data and likewise leveraging the downstream workflows, tools, and environments that are available. In addition to genomic and molecular data, what we've learned time and time again is that paired with biology, at the molecular level, you can really empower discovery by bringing in clinical and phenotypic data. This has been a fairly steep challenge in the genomics context, with really two communities operating independently, the clinical informatics teams oftentimes working independently from the bioinformatics teams. We sought to really try and create a uniform environment for this by, in Kids First, and again, as part of interoperability efforts across the NIH, implementing FHIR-based resources and including then OMOP interoperability. And then more recently have engaged the Tufts team and TGen once again to look for ways that really leverage what they have been doing in the context of the canine clinical trial landscape and the deployment of OMAP in ways that can further intersect across not only genomic and molecular data harmonization, but also clinical data harmonization. And we hope that bringing these to bear together, and as you can tell, some of these are still fairly nascent, just beginning efforts, but bringing this together across new data generation, existing data sets, platforms that can interoperate with new resources that then span both the molecular and clinical context, we can open the door for accelerated discovery on behalf of both children and dogs. And brain tumors for us really provide a unique unmet need for both drug development, the modeling of the immune system, the addressing of the blood-brain barrier, and the development of new therapies. And we hope, really sincerely, to expand these collaborations, leverage existing and shared resources, and ways that we can interoperate across these efforts on behalf of accelerated discovery. Thanks so much for your time and for listening, and I look forward to really partnering in any context, pediatric, canine, or otherwise. And everything I just spoke about is available presently and open for use and access within the Kids First data resource. So thanks so much, and I look forward to the rest of the meeting.
All right, that was really wonderful, Adam. Thank you so much for your presentation. Something I forgot to mention at the beginning is if those of you in the audience, you have questions, please type them in the Q&A bar, and we'll make sure when we get to the discussion section that we ask those questions. So if you've got thoughts right now, things you'd like to be able to ask Adam, please put them in the Q&A section, and we'll answer them when all the presentations are complete, and we'll have hopefully a little bit of a panel discussion. So I'd like to now introduce our next speaker. It's my pleasure to introduce Dr. Connie Sommers at the NCI in the Division of Cancer Treatment and Diagnosis. She is in the Developmental Therapeutics program, and she's the program director in the Immunotherapy Oncology branch. And Dr. Sommers has been working for a number of years in immunotherapy, and in particular in canine immunotherapy. And you're going to get a chance to hear some of her work directly from her. Dr. Sommers.
Good afternoon. I'd like to thank Ulrike and the organizers of this meeting for inviting me to speak about NCI resources for canine comparative oncology, and specifically about the Integrated Canine Data Commons. I work at the Division of Cancer Treatment and Diagnosis in NCI, and we support cancer research in canines to define the utility of using dogs as a translational model for human cancer therapies. And we've done this at 3 different levels. First, in 2016, at the genetic level, we funded cancer center grant supplements to investigate the genetic basis of multiple canine tumor types. And then in 2017, we funded 5 research project grants along with a coordinating center in conjunction with the Cancer Moonshot Project to initiate cutting-edge canine immunotherapy trials and correlative studies. And I'll mention these 2 efforts briefly. Then I'll concentrate more on the main event for comparative data analysis, which is the ICBC, which we created to provide a publicly available database that can be accessed for comparison of canine cancer data with human cancer data. So first, the supplements to cancer center grants were awarded in 2016. There were 8 one-year supplements to support research in canine cancer research via collaborations between NCI designated cancer centers and veterinary medical colleges. The kind of research that was supported was mostly for sequencing. This was whole exome and RNA sequencing, and each supplement was to sequence at least 25 canine tumors and their normal controls, and there were a variety of tumors that were investigated: B-cell lymphomas, gliomas, osteosarcomas, melanomas, bladder cancer, and mammary cancer. So these were initiated a few years ago. They're starting to bear fruit. These are just two examples of papers. On the bottom, you can see from Debbie Knapp, a paper that utilized data that was generated from the P30 supplement in bladder cancer. And then on top, from Roel Verhaak, there's this was partially supported by the P30 supplements, looking at canine and human gliomas. And what he found was that the canine gliomas were most similar to human pediatric gliomas. So these are some of the studies that are just starting to come out that are partially based on these supplements. So the second effort was Cancer Moonshot, the canine immunotherapy trials and correlative studies. So this network calls itself Precinct, for Pre-Medical Cancer Immunotherapy Network for Canine Trials. It's been a collaboration between the National Cancer Institute and the Coordinating Center at the University of Pennsylvania. And then there are 5 different research groups, which are seen on the right-hand side of the slide here. And you will recognize that some of these investigators have been speakers at this conference. Just as an overview of the kind of research that's being done and the kind of therapies that are being utilized, here you can see different investigators that are looking at brain tumors, lymphomas, osteosarcomas, and melanomas. And then in terms of therapies, there are tumor microenvironment modulators, there's cytokine therapy, autologous cell therapies like NK cell therapy. There's a vaccination approach with tumor lysate, checkpoint inhibitors like a novel checkpoint inhibitor, CD200, oncolytic virus therapy, and then a lot of combinations of these different therapies. So in terms of, where was this data all going to go? The genetic data, the clinical and correlative data from Precinct. We wanted these data to be publicly available. So the DCTD charged the Frederick National Laboratory for Cancer Research to build the Integrated Canine Data Commons, or ICDC, which is a cloud-based repository of canine cancer data. It was established to further research on human cancers by enabling comparative analysis with canine cancers. The data in ICDC are sourced from multiple different programs and projects, but they're all focused on canine subjects. The official launch of the ICDC was August 26, 2020, which is also National Dog Day. And you'll be seeing this website several times during my talk, caninecommons.cancer.gov. That's where data can be accessed from ICDC. So what types of data can be submitted? So of course there's descriptive case data like breed, sex, diagnosis, stage of disease. There's also genetic data like genomic and transcriptomic data, of course. Clinical data like clinical responses, adverse events. We'll also house imaging data like pathology, histology, x-rays, PET scans, and other scans. Immunophenotyping data can be submitted. That can be like ELISAs or links to flow cytometry. And then, as I'll describe, we're making the system flexible so that there are other kinds of data that you would like to submit. We'll also be able to accommodate that. ICDC is part of the larger Cancer Research Data Commons, and this is a large ecosystem, and I'll concentrate here on the different commons in the system. You probably are familiar with the Human Genomic Data Commons, and then some newer ones like there's an Imaging Data Commons, Clinical Trial Data Commons, and of course what we're focusing on here, the Integrated Canine Data Commons. The ICDC data model is based on a Neo4j graph database. And as I just mentioned, there's a flexibility to add new nodes and properties, so we can put in new types of data if that's indicated. It allows for both clinical trial type studies as well as molecular characterization type studies, and users may search across the entire system using a faceted search or through the API. The data model is available at this link listed here, and also a visualization of the nodes and properties is available at this link listed on this slide. In terms of workflow, the data that you start with will be curated and harmonized and eventually transformed so that it can be compared from study to study without any trouble. So then the transformed and finalized data goes to the user interface, which is the website. And this is a screenshot from the website. So I'd like to focus your attention to the left. You can filter by study type, by breed, by diagnosis. So you can really pull out what kind of data that you want. If we look, for example, here at breed, you can also hover over different sections, and you can see how many cases of a different breed we have and decide exactly what you would like to focus on for your analysis. And then the analysis will be done outside the ICDC, but there will be links and helpful information to help you get started with that analysis. We will be linked to a number of different ways of helping you analyze your data. For instance, the Cancer Genomics Cloud by 7 Bridges can help with an analysis of sequence data in many different ways. For what kind of queries is ICDC a useful resource? I've just come up with a couple of examples here. Of course, this is not inclusive. Hopefully you can think of a lot more, but if you look at a specific cancer type, say glioma, if you want to look at a certain mutation in a certain genetic region of interest, you can say, is that specific for a group of breeds like boxers, bulldogs, and terriers versus all other breeds, or compare individual breeds, compare really whatever group you want for a mutation or a region of interest that you're interested in. Another application might be if you want to look at a particular disease type like canine mast cell tumors and ask, are they more similar to human melanomas or to human mast cell tumors or to lung tumors? As long as you have the canine data and the human data that you can access to do those comparisons, that would be somewhere that you could go with this. In terms of therapy, does treatment with drug X in cancer type Y show similarities in dogs versus humans? So if you have the same drug or a different drug and you can find the data for the humans, you can compare, for instance, clinical responses. You can compare if you have histology or immunophenotyping, T-cell infiltration. You can compare tumor microenvironment composition. You can compare different pathways of gene expression. Really, whatever you can find, hopefully you can compare, and this will put us farther along the track on determining how good the dog is as a model for particular human cancers and how much we can do in dogs that will inform therapies for humans. But really, we're interested in what kind of queries would you like to use the ICDC for. So hopefully in the panel discussion part, you can let us know what you're interested in, what kind of questions you would like to ask, and then we can try to make sure we can accommodate that. Here's a list of some studies that have already been submitted to ICDC, and there are a number of different disease types, as you can see, lymphoma, glioma, bladder cancer. One thing I'd like to point out here, if you would like to submit data, but you're not ready for it to be publicly released, you can go ahead and submit it. and we can embargo the data until publication or until any date that you wish. So we're trying to be pretty flexible with this system. If there's something that you need, please ask, and hopefully we can accommodate you. So how do you access data in ICDC? Again, here's the website, caninecommons.cancer.gov, and how do you submit data? You go to the same website, and here's the link for how to submit data. And if you have questions about submitting or any other aspects of the ICDC, please email us at icdchelpdesk@mail.nih.gov with your questions or comments or suggestions. Lastly, I'd like to acknowledge a number of people who have helped in the ICDC effort. First, Toby Heck from the NCI, who helped initiate these canine research programs at NCI and is a guiding force all along for all of these efforts. Amy LeBlanc, who's in the intramural NCI program, who is a great advisor on all veterinary issues to us. Anju Singh, who's in the ImmunoOncology branch, where I am also. Erica Kim, who is at CBIT and helps us with the data part of these efforts. Matthew Byers is from Frederick National Labs, and he's giving a poster at this meeting, so please go to his poster presentation, and he can help you with some of the more technical aspects of ICDC. Also, John Otridge is from Frederick National Labs and oversees the ICDC and other data efforts, and he will be in the discussion following this session. So he can also be available to answer your technical questions. And lastly, I'd like to thank Debbie Knapp, who's the chairman of our ICDC steering committee, and you'll be hearing from after my talk. And really all of the members of the ICDC steering committee from the extramural community. We're really grateful for all the help, input, advice that you've given us to try to make this a user-friendly and useful resource for the extramural community. I'd like to thank you for your time and attention, and I look forward to the panel discussion time and your questions. Thank you.
All right, thank you, Connie. That was a wonderful talk, and obviously I had a little bit of a brain glitch that was all about the ICDC, and I apologize for the wrong introduction there. Again, I want to encourage everybody to put their questions down in the Q&A, so we have your questions. Otherwise, you'll be stuck listening to my questions, and I'd much rather ask yours. So we have one more presentation from Dr. Deborah Knapp. And as you heard from Dr. Sommers, she is the chair of the ICDC steering committee, but of course she's much more than that. She's a Dolores L. McCall Professor of Comparative Oncology and the director of the Purdue Comparative Oncology program. So she's a professor at Purdue University, and she is a Doctor of Veterinary Medicine. So with that, I would like to introduce Dr. Knapp, and you will get to hear some of her thoughts on the power of comparative oncology.
Good afternoon. The title of my talk is Genomic Characterization of Canine and Human Invasive Bladder Cancer, Lessons and Opportunities for Adult and Pediatric Cancer Data Integration. I'm Debbie Knapp. I appreciate the opportunity to participate in this meeting and share some of our work with you. If we think about how are we going to make advances in the management of cancer in dogs and in humans, we have to make progress on three fronts. The first is on better therapies. We need more targeted drugs, immunotherapies, drugs we honestly haven't even conceptualized yet, and combination therapies. We also need to be able to select the patients that we know will respond best to specific drugs, and we ideally need to make progress on cancer prevention. Although I'm going to use an example of an adult cancer, that is invasive bladder cancer, these processes apply to pediatric cancer as well as to adult cancer. And in both adult and pediatric cancer, we need highly relevant animal models because there are not enough human patients and not enough money to test all of the new strategies that we may want to evaluate. It's been, I think, 2 years ago, I attended a human bladder cancer meeting, and one of the speakers put a slide up, Noah Hahn from Johns Hopkins, a very compelling slide. And he outlined all of the new strategies that we should test in humans. And then when he added up the number of trials, it was going to be something like 36 new trials. And there just aren't enough resources to do all the trials that we want. And that makes preclinical or pre-human work in animal models very, very, very important. So dogs can fill this niche because of their relevance to the human condition. And related to the topic of the discussion here, genomics really are key. The example I'm going to use is invasive urinary bladder cancer. It's also called invasive urothelial carcinoma. It's also called transitional cell carcinoma, or TCC. And this disease in dogs and humans is very similar in the clinical presentation, in the pathology, and in aggressive cancer behavior. It invades into the bladder wall, and at least half of patients, both dogs and humans, it spreads to distant sites in the body. This cancer in dogs and humans is also similar in molecular features. And specifically, I'm going to focus in on what's called molecular subtypes. These subtypes are defined by RNA-seq or by expression data. They were first identified in human breast cancer, but they're also present in human bladder cancer. And the reason we care is these subtypes affect how the cancer will behave and how it will respond to therapy or not. Across several studies in humans, really worldwide studies, there are consistent findings of two major subtypes. They're called luminal and basal. And the luminal tumors tend to be less aggressive and tend to respond better to targeted therapies, whereas the basal tumors tend to be very aggressive with early metastasis, frequent metastasis, but interestingly, they may respond better to chemotherapy and respond better to immunotherapies. And so these subtypes are really important in being able to move toward personalized or individualized care. Well, these subtypes exist in dog bladder cancer, and I'm going to use this heat map to demonstrate the point. The way this map is set up, each column represents one case, and each row represents one gene. And the expression of the gene goes from green, which is really underexpressed, to red, which is highly overexpressed. The way the heat map is set up, the columns under the green heading are basal tumors, and the columns under the yellow heading are luminal tumors. The gene set that was used to create this heat map is the same gene set that classifies human bladder cancer as luminal and basal. And I think you can appreciate how the color patterns really do distinguish these subtypes in the dog cancer as well. One of the other things that's really important about these subtypes and really fits into the huge wave of immunotherapy that we're all seeing is that the immune component of the tumors differs between the luminal and the basal subsets. So in this heat map, again, each column is a case and each row is a gene. And the green columns are the basal tumors and the yellow columns are the luminal tumors. Except the genes in this heat map are a panel of genes that is used to classify human bladder cancer as either immune hot or immune cold. And by immune hot, I mean that it's inflamed, it has infiltration of T lymphocytes, it has pro-immunologic signatures. On the other hand, immune cold means the tumor is not infiltrated by immune cells and it has immune suppressive features. In this heat map, the way we've set it up is that the red boxes are going to represent pro-inflammatory patterns, and the green boxes are going to represent the immune suppressive patterns. And I think it's very, very obvious that the basal tumors, again, those under the green headings, are immune hot, and the luminal tumors are immune cold. So now this offers us the opportunity to actually select tumors for the study that we want to do. So if you want to study a strategy to convert immune cold to immune hot, and you'd want to do that because the immune hot tumors are much more responsive to a variety of therapies, you could study the luminal tumors in dogs. On the other hand, if you're already to go with an immunotherapy, you would want to pick the tumors, the basal tumors, because they are already primed to respond to an immunotherapy. So Connie, in the talk right before ours, gave a really good overview of the NCI's Integrated Canine Data Commons, or ICDC. We are depositing 2 bladder cancer data sets into this common, and one of them builds on this luminal basal subtype work. And I can tell you that with approximately twice as many cases as the ones I've just shown you, the results are even more compelling. The subtypes are definitely apparent, and the immune infiltrates within those subtypes, the data is just even more compelling than what I've just showed you. So again, this gives us the opportunity to begin to study personalized care by selecting the patients, i.e. the dogs, that have not just the subtype that we want to study, but the immune type. So if I could go back now to the title of the talk, I'm supposed to cover lessons and opportunities. So let's do that. And let's start with opportunities because that's what I get most excited about. So this is the same slide I showed you earlier. This is where we need to make progress on the 3 fronts. And I am absolutely convinced that dogs are going to help us do this, and that genomics is going to be a key piece to this, because we have the opportunity in dogs to study these new therapies. We have the opportunity to study personalized strategies such as the subtypes, that we can build on that with much more with the genomic signatures. And based on the high breed associated risks for bladder cancer in dogs, we have unique opportunities for prevention research. But that's for another day. In terms of the lessons, I'd like to mention 2 things. These reflect my thoughts on this whole field recently. First of all, we need all models. Even though I am a champion for the dog models, we need everything. We do need the experimental models. They provide the opportunity to do studies in a very tightly controlled environment. They offer opportunities to do mechanistic studies. So we need those. I am in no way suggesting that we do away with any of the models. Rather, I think dogs will complement those because the tumors in dogs are so much more similar to those in humans than some of the rodent models. So my second lesson is genomics are really key because they will allow us to use dog tumors for a variety of different reasons. So one of the interesting things about dog bladder cancer is that the majority of cases express a mutation in the BRAF gene. It's fascinating. It's the same gene, the same position on the gene, and the same substitution that occurs in 8% of all human cancer. And so this means that people that are interested in this BRAF mutation may tap into dog bladder cancer for their studies. This would include people studying metastatic melanoma in humans because that's where BRAF mutations are perhaps most important. So now not only do we have a bladder model, but we have a model for other types of studies. On the other hand, if you want to study the subtypes related to bladder cancer, the dog is ideal for that because you have the subtypes. And what I think is interesting is that even though BRAF mutations are common in dogs, they're not that common in human bladder cancer. In the more advanced, really aggressive tumors, yeah, we see it more. But across bladder cancer in humans, they're not that common. And so I think it's fascinating, though, that the subtypes are still persisting, even though different drivers may set it all in motion. So I'm really excited about where the field of comparative oncology is going, where the genomics is allowing us to go. And I think it's an ideal time to be working in this area of research. And I'd like to thank you for your attention. On this slide, I'm showing some of the dogs that have participated in our program. All of them have bladder cancer, except the one in the upper right, and that's my own dog. And I also have to say, I feel very, very fortunate to work with so many very, very talented people. I want to point out Dr. Dewan, who did a lot of the bioinformatics work and led a lot of the subtyping studies in our group. And at this point, I'll be happy to pause and take any questions that you have. Thank you.
Wonderful. Thank you so much, Debbie. That was a great talk. And I loved you ending on pause. I do have so I'm going to start with more of a philosophical question, then I'll get into some other questions. Thinking about you mentioned preclinical models, and that evokes to me really the either cell models or rodent models, and yet the canine models are so much closer to human, right? Because they are spontaneous mutations, they are spontaneous cancers. Is it fair to call them preclinical models? Should we have a different name for them?
I'm not sure. Can you all hear me?
Yes.
Okay, cool. So, great question. I do think we should call them preclinical models. I can certainly understand. Coming at it from different angles, though, and they definitely are a unique preclinical model compared to the others that the scientific community is used to working with. Perhaps others on the panel could weigh in on that question as well.
Yeah, I can speak a little bit about this. This is Adam. Can you guys hear me?
Yes.
Yep. So, one of the things that, and one of the reasons I focused on brain tumors is because one of the biggest challenges, for example, that we face in brain tumors, and this is true for both adults and pediatrics, is actually drug delivery. And so, the reason I bring this up is because canines provide a model in which we can assess drug delivery that overcomes the challenges of the blood-brain barrier, for example. So even as you start thinking about the context of models, whether it's the mutational context or the organ-specific context, the other is even just the implementation or applicability context. And there is no other quote-unquote model, whether it's rodent or others, that can fully recapitulate the challenges we face, for example, in blood-brain barrier overcoming. So I think that is certainly one of the ways that we think about the canine space. And alternatively, I guess we could also think that humans are the preclinical models for canines. That's certainly true for BRAF, for example.
That's a great point. And actually, someone made a comment in the Q&A that to call them true disease models versus just preclinical models. Yeah. It does bring up a different question. And again, Debbie, you were talking a lot about BRAF. So this is where my knowledge of canine cancer isn't as great as I'd like. So does things like vemurafenib, which are so effective in melanoma, do they work in dogs that have these BRAF mutations, the same BRAF mutation?
We have just completed a clinical trial of vemurafenib in dogs with bladder cancer. And the answer to your question is yes. The remission rate is approaching 40%, which is, as a true single agent, that's better than the other true single agents that we have used. So absolutely, they have activity and acceptable safety. And one of the data sets you'll see in the ICDC as soon as we can unembargo it, once it's in print, or certainly in press, is we have genomic data from cystoscopic biopsies before treatment, during treatment, and at relapse. And we're able to follow the changes in various pathways, which is very informative. And I think one of the more exciting pieces of data coming out of the study is that we're seeing definite changes in the immune state, i.e. tumors are switching from immune cold to immune hot when the tumors are responding, and then they're going back to immune cold when the tumors relapse. I don't have all that information right in front of me right now, but we are happy to share it with the scientific community by publishing it and putting it in the commons, and hope others will mine it for additional information as well.
Well, great. That's wonderful. It does bring up another question, a follow-on question. Sorry, Debbie, I don't mean to pick on you.
It's okay.
Thinking about BRAF, again, there are other human cancers that have BRAF mutations, V600E, that aren't responsive to vemurafenib, like most colorectal cancers, for instance.
Right.
It's actually pretty exciting to think about now, looking at relapsed dogs and seeing what pathways get changed. And does that correspond to the normal state of a colorectal cancer? And does that give us a different key into the biology of colorectal cancer?
I think it can. One of the things we've done is to look up the various pathway alterations that occur in humans that do not respond to BRAF inhibitors, or those that initially respond and then fail. And looking at pathways and looking at specific genes, there's a lot of overlap between what we're seeing in the dog tumors and what's reported in people. But there are also some additional changes that haven't yet been reported. So I think your question is intriguing in that the response we see in dogs is probably not quite as good as what's seen in human melanoma. And it does appear better than colorectal and thyroid. So I think the dog tumors will actually end up being somewhere in the middle. And hopefully that will be even more, provide even more information that will help us understand why do these drugs fail when they fail? Because when they respond, people that respond have incredible, dramatic responses. I mean, people with really advanced cancer can have durable, complete remissions. And if we could help figure out how to make everybody experience that, that would be wonderful.
Great. We have a follow-up question on that point. And this is from Christiana Oreston. Related to the BRAF question, I'm wondering if the group can comment on the inclination to study canine cancers in a histology-directed manner. Should we be looking at tumors as all comers and looking how they match to human genomic genetic data after the fact?
I think we need to do something.
Debbie, you want to answer that, or somebody else wants to answer that? No.
My response briefly is I think we need to do both. I think we are starting out, most of the work to date has been done on histologically defined tumors, but I think we now have the opportunity to define the tumors based on the genotype. And so I think we need to do both, but please, others weigh in as well.
And it might be different for canines and for humans, the histologies and the connection to the genetics. So I think, like you're saying, we just need to do them both and find out.
Mm-hmm. Sure.
And I think there are a couple of, coming from the pediatric space where, kids don't get cancer because they smoke or they overeat or they're out in the sun too long. And I suspect similarly, there's an element of predisposition in the context of canines that really points to mechanistic underpinnings that potentially intersect with development or epigenetic control, where you do have really tremendous capacity to penetrate some of that shared biology that might be underpinning, at least in our case, pediatric and canine cancers. So from that perspective, I think there's a ton of potential. And then just to add another couple of points, oftentimes we don't get the privilege of actually analyzing tumors after therapy in the human context, or when therapies fail, what does a tumor actually look like after that? it's very rare to have sequential samples in this context. And again, this is something that can be, in a histological sort of agnostic manner, really powerful to really think about, before treatment and after treatment, how tumors respond to therapy independent of histology, for example. Just a couple of thoughts to consider.
One of the things I had on my slide and forgot to actually talk about was the Presync group is putting together a nanostring platform that will have immunotyping, and there'll be some genes in there for subtyping as well. And one of the reasons that I'm particularly excited about that is it is a way to standardize how you define the immune state. And one of the ways that our group plans to do it is to determine if it works in urine, because if it does, then that gives you a tremendous opportunity to follow a lot of the information about the tumor over the course of treatment at multiple time points. So Nikki Mason and others have taken the lead on that, and we're really looking forward to test that panel. We have a smaller panel our groups put together that we know works on urine, but I think the one coming out of Presync will be even more powerful.
Yeah.
Great. Thank you. So we have another question that's come in. Now I'll read it. It's from Carol Robertson-Plough. The canine human bladder cancer work that Dr. Knapp has conducted is a great example of leveraging target-focused comparative research. Moving forward, how is the best way, or what would the best way be for a researcher that wants to leverage the canine ICDC and the human CBTTC for starting a new investigation into relevant targets in dogs? It's a little bit of a complicated question, but really it's how do we take advantage of some of the infrastructure that we've invested in to enable new therapeutic research?
Yeah. So maybe I can start and others can chime in. I think this is one of the most exciting things that's been happening at the NIH and NCI level. For us in the pediatric community, for example, or the brain tumor community, we recognize that we don't have enough investigators that care about pediatric brain tumors. So many of our efforts around interoperability are to drive chance discovery. And what I tried to highlight in my talk is that some of these emerging platforms and their implementation provide for that interoperability, where a user who might want to create a cohort of data between brain tumors and canines can bring them into a common workspace and do an analysis, or a portal environment can bring process results from 2 different studies and support that analysis. And I think the more we as a community start doing those types of efforts, the more sort of the mimetic nature of science will take over, and others will do it because it will drive that chance discovery. For example, Rolls, really phenomenal studies showing that canine gliomas share some biology with pediatric brain tumors is a perfect example for the kinds of studies we want people to do, to then bring those 2 together, those 2 data sets, harmonize the bioinformatic approaches, deploy them in common environments, then expand that to bladder and any other cancer. So my hope, even after today, is that those types of efforts can take shape, and we're certainly happy from our side of the CBTTC or brain tumor cohorts or Kids First to support these types of efforts, or working groups who want to look at some of these comparative biology questions and some of these platforms.
This is John. Just to add on to what Adam said, is that in his presentation, it's very clear that you can take the data and analyze it in Cavatica. Well, Cavatica is built on 7 Bridges Genomics, which is based on Amazon and the ICDC. You can go and find cohorts there, and you find cohorts of interest, and you can bring them in, and you can combine them in 7 Bridges and analyze them, and you can bring your own data in there also. So I don't think that there's anytime soon we're going to have one system to rule them all for everybody. It's all about being able to enable interoperability. The harmonization of the data is a critical path in this, as is the community aspects. And I think this aspect of, Adam did mention a little bit about communities. There's multiple communities. There's a canine community, and helping the canine community understand, when we gather data, can we gather it in a way that makes it more interoperable downstream, not just for our own needs here? Can be even more value extracted from the samples that have been carefully gathered? And I think these are the big changes that we're seeing, and the cloud enables us all to come together in ways that we haven't before, across not just genomics, but also proteomics and imaging. So I think it's really exciting with the threshold of so many of these pieces converging together, that we're going to have an explosion of discoveries in the next few years, with the funding on the grants as well as the funding on the infrastructure.
Thank you, John. So we're just about out of time. Actually, we're a little over time. Debbie, would you like to make a comment? And then Connie, I will let you close us off, since you had the middle spot in the presenting.
I'm not sure I have anything else to add. Thank you.
Yeah, and I don't really have much to add too, except I really want to thank the audience and thank the participants, and keep the suggestions coming in. It really is an exciting time. Thank you.
All right. Well, thank you all. It was great to be here, and hopefully next time we'll actually be in person.
Yeah. Thank you, Warren.
Thank you, everybody.
Okay. Take care.